Great.
So we're going to start off with 3 presentations. So we're going to be about 5 minutes each. So have questions and they go on for the next 3. So this will be questions. And we're going to start off with Dr. Lucas from the CEO conference. And he'll be talking about the next topic and he'll tell us about microbiome. It's a hot topic these days. And patients with metastatic RCC regarding response to injectables.
Thank you. So good afternoon, everyone. It's a pleasure to be here. I'm Luis Mesa. I'm a fellow working at the Medical Oncology Department at City of Hope. And today to present the results of our study, was just presented the taxonomic and functional assessment of the gut microbiome in patients with metastatic RCC in regards to response to immune checkpoint inhibition. I have no disclosures. So this is a very interesting time for RCC, as we've noted for the talks this morning. And we've seen a set of critical data sets regarding the use of ICIs in many treatment settings for RCC. But one of the questions that always comes up is, what's the role of gut microbiome in this type of treatment? And we've seen a lot of growing body of evidence suggesting that there are certain bacterial species that have been associated with response to ICIs in metastatic RCC, but what we don't quite know is what are the mechanisms that drive this association and this response. So in this study, we examined a taxonomic and functional profile of the gut microbiome in patients with metastatic RCC treated with ICIs. So for this, we devised a study in which we accrued patients with metastatic RCC prior to initiation of ICIs. Then we collected data on demographics and clinical information, and we collected stool samples at baseline and after 12 weeks of treatment. We then divided our patients into responders and non-responders by RECIST criteria and used human treat to profile the abundance of microbial metabolic pathways in the samples. So we enrolled a total of 31 patients. The median age was 61, most of them male, and most of them clear histology, 71 and 90% respectively. IMDC risk, intermediate and poor were seen in 61% of patients, and nivolumab was the treatment of choice for 77 patients, and nivolumab for 23% of patients. 11 patients, or 35%, responded to treatment. For bacterial composition results, we see really key differences between both of our cohorts, both in responders and non-responders. So we saw, for example, that in responders, there was an increased relative abundance of Bacteria subbatus, Artorchis, and E. ramulus. There are bacteria known to produce short-chain fatty acids. Whereas in non-responders, we see Clostridium bolte and Clostridium valenciensis, which are bacteria that we know produce carcinogenic compounds such as N-nitrosa compounds. Yeah, these are not the latest slides, but it's fine. And so for functional pathways, we also saw a couple of big differences. First, in glycogen degradation and sucrose degradation for responders. And then in the non-responder group, we saw increased relative abundance of pyruronate degradation and galacturonate degradation. These are 2 pathways that are associated with pectin degradation, which has been hypothesized to influence in pro-inflammatory cytokines, as well as the expression of short-chain fatty acid transproteins. So in conclusion, we believe that the taxonomic and functional profile of the gut microbiome could serve as a factor distinguishing responders from non-responders in patients with metastatic RCC receiving ICIs. The fecal abundance of species known to produce short-chain fatty acids was increased in patients who responded, whereas bacteria known to produce carcinogenic compounds such as N-nitrosa compounds was seen in increasing abundance in patients that didn't respond to ICIs. We believe that incorporation of taxonomic and functional profiling should be investigated in bigger studies, and we hope that one day we can use microbiome-based biomarkers to selectively identify those patients that are going to benefit from ICIs. I'd like to thank the patients that participated in the study, my mentor, Dr. Samantha Pell, our team at City of Hope, and all of you for listening to my talk.
Good job.
All right, wonderful job. Our next speaker is Dr. Nishita Tripathi, and she's going to be talking about the genomic and transcriptive predictors of response to treatment with Ipi/Nivo in patients with metastatic RCC.
Good afternoon, everyone. I am Nishita Tripathi. I am a afternoon, everyone. I am Nishida Tripathi. I'm a postdoctorate fellow working at Huntsman Cancer Institute, University of Utah. And I would like to thank KCRS for giving me this wonderful opportunity to talk about the results of our study titled Genomic and Transcriptomic Predictors of Response to First-Line Treatment with Ipilimumab and Nivolumab in patients with metastatic clear cell RCC. I have no disclosures. The combination therapy of ipilimumab and nivolumab is one of the standard of care treatments for patients with metastatic renal cell carcinoma. Previously, genomic and transcriptomic biomarkers have been explored and have been found to correlate with response to immunotherapy or immunotherapy-containing regimens in metastatic renal cell carcinoma. With that context, through our study, we aim to explore genomic and transcriptomic biomarkers associated with disease control rate for patients treated with EP-nivo therapy having metastatic clear cell renal cell carcinoma. So for our study, we included all patients with metastatic renal cell carcinoma, having clear cell histology, having intermediate to poor risk IMDC prognosis, and were treated with upfront EP-nivo therapy. And all of these patients had baseline tumor-based comprehensive genomic profiling done prior to the initiation of ipi-nivo therapy. We also divided the patients into two cohorts based on the disease control rate observed. And as you can see, there were two cohorts, the disease control cohort and the no disease control cohort. Based on the available DNA profiling data, we had total 49 patients, of which 33 could achieve disease control, while 16 could not achieve disease control. We had 14 patients with available transcriptomic data, of which 10 could achieve disease control and 4 could not. We looked at the clinical and demographic characteristics to see any difference between the two cohorts, and we found that the IMDC risk prognosis was significantly different between the two groups. Further, we looked at the frequency of genomic alterations in between the two cohorts, and we found that VHL and PBRM were the most frequently altered genes in both these cohorts. But in our study, none of these alterations were significantly different between the two cohorts. Further, we did a genomic expression analysis, and we found that the disease control cohort had significantly higher number of differentially expressed genes, which is close to 40 genes, compared to the no disease control cohort, which had only 11 genes. Further, we looked at the pathway which were upregulated between the two cohorts and found that the disease control cohort had upregulation of pathways involved in cellular proliferation, such as E2F, the G2M checkpoint, the MYC target, and the estrogen late response. And in contrast, the no disease control cohort had TGF-beta signaling. Our study found that the disease control cohort had upregulation of pathways involved in cellular proliferation. Cellular proliferation in itself as a cellular event acts as a stimulus for recruitment of both pro-tumor and anti-tumor cells in the tumor microenvironment. And our study suggests that this could modulate the activity of ipi-nivo in these patients. Compared to the no disease control cohort who had increased TGF-beta signaling, the pathway is involved in the production of immunosuppressive cytokines, and these cytokines enable the tumor cells to evade the host immunosurveillance in these patients. Further, the limitations of our study includes the retrospective nature and the small sample size, and we feel that the results of our study are hypothesis generating and needs to be externally validated in a larger data set. In conclusion, our study highlights the importance of identifying genomic and transcriptomic biomarkers, which could help identify patients who may derive a greater benefit from the ipi-nivo therapy and may further help in treatment selection and prognostication. At the end, I'd like to thank my mentors, Dr. Neeraj Agarwal and Dr. Paul, for their unwavering support and guidance throughout this project. Also, not to forget the wonderful team at the City of Hope and Huntsman for making this project possible. Thank you so much.
All right, last but not least, we have Tejas Jamal from MD Anderson talking about, which we were just learning a little bit of tidbit in our last talk about tertiary lymphoid structures and clonal neoantigen load. So good segue from before.
Today I'll be talking about exceptional response to immune checkpoint inhibition in clear cell renal cell carcinoma. Immune checkpoint inhibitors have been transformational in CCRCC. However, only a small minority of these patients have exceptional responses, which are marked by deep tumor regression and prolonged survival benefits. In order to gain mechanistic insights, we asked what are the genomic and transcriptomic correlates of exceptional response in CCRCC. So we define exceptional response as patients who have complete response and a progression-free survival of over 12 months, or patients with a partial response, tumor shrinkage of over 50%, and a progression-free survival of over 24 months, or patients with partial response and progression-free survival of over 36 months. We also define intermediate response as all responders who are not exceptional responders and PD as progressive disease. So we obtained baseline samples from 2 treatment-naive advanced RCC cohorts. The first cohort was treated with a combination of 2 checkpoint inhibitors, and we obtained exome data from this cohort. The second cohort was treated with a combination of checkpoint inhibitor and VEGF inhibitor, and we obtained both exome and RNA-seq data from this cohort. So in order to look at the genomic correlates, we looked at all the previously described genomic markers of response, and we did not see any association with exceptional response. However, we see that clonal neoantigen load, which is defined as the total number of predicted neoantigens which arise from clonal mutations, is significantly associated with exceptional response as compared to both IR and PD. Notably, we do not see this difference when we look at all responders versus progressive disease, suggesting that clonal neoantigen load is associated with exceptional but not overall response. We also see that higher clonal neoantigen load is associated with improved progression-free survival outcomes, and a multivariate analysis shows that clonal neoantigen load is an independent predictor of progression-free survival. We then looked at the second cohort, and the genomic analysis did not show any significant associations. We looked at the RNA-seq data, and we see that in exceptional responders, there is an enrichment of pathways associated with intratumoral humoral responses, such as signaling by the B-cell receptor pathway. An immune deconvolution analysis showed that in exceptional responders, there is an enrichment of plasma cells, CD8 T cells, and memory B cells. Recent studies have shown that in CCRCC, tertiary lymphoid structure formation is associated with improved outcomes and responses. And since B cell and plasma cells are a major component of TLS, we wanted to ask if this observed signature is driven by formation of TLS. And indeed, we see that in the exceptional responders, the TLS signature is much higher compared to both IR and PD. And also, there's a trend of higher TLS signature in IR compared to PD, even though it's not statistically significant. Thus, the TLS score is preferably associated with exceptional response as compared to intermediate response. And also, TLS signature is an independent predictor of PFS. Next, we looked at all the genes comprising the TLS signature, and we see 2 distinct subgroups. The first group is marked by an enrichment of these genes, and the second group has lower expression of these genes. The exceptional responders with a lower expression of TLS associated genes have higher clonal neoantigen load, suggesting there are 2 distinct pathways of achieving exceptional response. The first group, which has higher B cell, plasma cell, and TLS signatures, seems to be driven by mostly antibody-mediated responses. And the second group, which has lower TLS score and higher clonal neoantigen load, is mostly driven by T-cell associated cytotoxic activity. Thus, to conclude, we see that clonal neoantigen load is associated with exceptional but not overall response, and it is an independent predictor of PFS. And in IO plus VEGF, TLS signature is an independent predictor of PFS, and there are 2 distinct subgroups, one with a high TLS expression and another with a lower TLS expression but higher clonal neoantigen load, showing that there are 2 distinct pathways of achieving exceptional response. I'd like to thank my mentors, Dr. Sachet Shukla, Dr. Tony Shoeri, and Dr. David Braun. I'd also like to thank all my collaborators and most importantly, patients and their families.
Any questions? I actually, as a clinician for you,
Yeah.
yes, you know, you think about immune-related adverse events specifically they can be related to high responders or low response. Did you find any correlation with those in your study about patients having a grade 3 or 4 IRE versus none in those different
So in our
cohorts?
study, we did not look at any adverse events. We just looked at the genomic and transcriptomic data. So yeah, we didn't look at that.
I mean, it would be interesting to see how they're responding to more antigens, what's going on in that biome as well, really characterizing that.
Oh, got the mic?
You have a mic there. There you go.
I'll try to use the mic, yeah. Do you have any insight into the nature of the neoantigens? Is it something that you sequenced, you did peptide mapping, do you know anything about their nature?
So we we predicted the neoantigens based on the somatic mutations. So for each patient, we have the exome data. So from that, we obtain the somatic mutations, and then we predict the binding of derived clonal neoantigens to the patient-specific HLAs. And we also looked at mutations which arise from clonal events, mutations which are present in all of the tumor cells.
So these were modeling of the neoantigens. I mean prediction of the neoantigen.
Yes.
Was not any direct interrogation of the presence of the neoantigen on tissue.
No. Okay. Yeah.
Ari?
Two quick questions. I'm curious, you know, we've been fascinated by the TLS, you know, when it was reported initially in the Nature paper a few years ago and subsequent work, but our pathologists rarely see them. I'm curious, given your expertise with Sabina and others at the Farber, if people have looked at the quote unquote TLS signature high tumors to actually see if they are actually seeing TLSs there or if it's picking up something else. That's my first question. And the second question regarding the clonal neoantigens is, did you restrict them to substitutions only, or did you look at indel neoantigens as well?
Yeah, so for the first question, yes, we are trying to validate the formation of TLS using immunohistochemistry. And for the second question, we looked at all the clonal neoantigens, both deriving from single nucleotide and indels.
Yeah, so Ari, we have started to look at the tertiary lymphoid structures, and so what we see in RCC, they're mostly located the interface between the tumor and the at the periphery of the tumor, and but we're also looking at the maturation state. So many lymphoid structures are not mature, but so it's a work in progress, but I think it's sometimes easy to miss their presence because they're not located in the center of the tumor, and sometimes I think maybe even genomic studies might not have included them for that reason.
We're going to do one quick question from the chat. What should the next trial be evaluating the microbiome and RCC?
So
easy question, right?
We actually have a lot of the research so far has been done in the IO only setting. So now that for example, being one of the favorite, I should say, first-line options, investigating TKIs plus IO in the setting of microbiome study, that'll be something interesting. Actually, something that we're doing at CDFO.
Actually, any data regarding the microbiome where you can look in the serum, perhaps, I don't know, antibodies against these bacteria, which will be a cleaner and you know, way to look at it rather than other collections.
So, yeah, I think that's understandable from a physician and a patient perspective, having to submit full specimen result. We've tentatively tried to look at that in the setting not of gut microbiome, but intratumoral microbiome, whereas, for example, TCR responses to bacteria identified within the tumor could be a signal of cleanliness, if you will, as opposed to just contamination because of the management of those carbon-embedded samples.
I've got a question for you. So we just recently saw that very elegant work from Rob Knight's group about the mycofungum. what are your strategies around tackling other sort of microbial, or not even microbial, I don't know if it's the right word, entities that could be present within the microenvironment?
So we tried to do it to some extent, looking at the microbiome, but unfortunately the yield of the analysis was not good enough for us to make conclusions based out of it. But definitely looking at viruses, looking at other things that might be embedded in the tumor and the gut, it's a way to go that we haven't just explored thoroughly.
Very exciting. All right, great. So we're going to shift on to the next three. If you guys want to step down, thank you so much. And we'll call up. Slides. I'm looking for a thumbs up and we're good.
Hot mic.
Thank you so much for giving me the opportunity to talk about our work. My name is Maurice Alcantara, as mentioned, and I'm a postdoc from City of Hope. So just to begin, we know that nivolumab and ipilimumab represent frontline therapy for metastatic RCC, and they aim to restore exhausted T cell function. However, we also know that there are a proportion of patients that don't respond for numerous reasons, including postulated to include, but are not limited to, the innate and adaptive immune status of patients, the activation of oncogenic pathways, and the tumor microenvironment. There are currently no established predictive biomarkers for patients and their lack of response to these immune checkpoint inhibitors or ICIs. And so in this study, we aim to investigate two parts. Firstly, in humans, investigate associations between immune alterations in lack of response to nivo-ipi, and then in mice, assess a novel drug combination that targets specifically the innate and adaptive immune alterations that we observed. So for the first part, we did a retrospective study, and 37 renal cell carcinoma patients treated with first-line nivo-ipi at City of Hope were identified. We analyzed blood samples taken at baseline and week 12 for various immune cells using flow cytometry and 30 circulating cytokines with Luminex. So here's what we observed in patients, and we had 21 that did not respond to nivo-ipi combinations. There were three main cytokines that were significantly upregulated: IL-6, IL-8, and IL-1RA. In addition, we also observed that there was a significant increase in circulating immunosuppressive T regulatory cells in patients that did not respond to therapy, alterations that were not evident in patients that responded to combination therapy. Then, utilizing Pca analysis in the same patients, we observed that there was high evidence for activated STAT3 or phosphorylated STAT3 in the same time points between baseline and week 12, and that this expression was mostly located within the myeloid cell cluster, located in purple. So just going back. So given that IL-6 is one of the main activators of STAT3 and the STAT3 pathway, we think that this may be relevant and correlated or connected in these patient samples, and they may play an important role potentially in resistance to nivo-ipi. So for the second part, we utilized the Renal mouse model of renal cell carcinoma, and we combined PD-1 blockade with a specific STAT3 inhibitor that targets STAT3 within myeloid cells, known as CpG STAT3 antisense. And then after treatment, we looked at the peripheral blood tumors and tumor-draining lymph nodes of these treated animals for various immune cells via flow cytometry, and then IL-6 with ELISAs. We found that combination therapy in red had better tumor growth control than PBS treated, IgG treated, or PD-1 blockade alone. We also found that underpinning this efficacy that in combination treated animals, there was an increase in CD8 positive T cells infiltrating the tumors, not evident in those that were treated with PBS or saline, the IgG control, PD-1 blockade. And then interestingly, we found changes within the innate immune cells, including the activation of dendritic cells in tumor draining lymph nodes, and then a shift from the pro-tumor M2 macrophages within tumors to the more anti-tumor M1 macrophages. So again, changes not evident in the PBS, IgG, or PD-1 alone. And then similar to humans analysis, we found that the circulating IL-6 increases in these tumors as the tumor progresses, but that we could significantly decrease IL-6 in combination treated groups. So just to summarize, non-responders to combination therapy have immunosuppressive Tregs and circulating cytokines IL-6, IL-8, IL-1RA, and high levels of activated phosphorylate STAT3, suggesting immune suppression, particularly within the innate immune cells. And then IL-6, IL-8, IL-1RA should be investigated further as potential biomarkers to nivel IPI. And finally, our animal studies suggest that combining a STAT3 inhibitor with PD-1 blockade may be able to overcome immune suppression in RCC and warrants further investigation. Finally, I'd like to thank my mentor, March, and then Dr. Paul and our team at City of Hope.
So this is advancing.
Our next speaker is Abhishek Chakara Baruti from Cleveland Clinic. He's going to be presenting on tumor cell states.
All right. The origins of this story really are back when I was still in Bill Kaelin's lab in Boston. And this is around the time when the Broad Institute started collecting a large collection of several thousand cancer cell lines. They call it the Cancer Cell Line Encyclopedia, or CCLE. And we asked a very simple question over here. What would be dependencies that would be lineage specific? In other words, can we compare what genes a kidney tumor would depend on much more than all other lineages? And this analysis led us to a number of genes. It was interesting to see that some kidney lineage master regulators like HNF1B, PAX8 were leading in this list, which gave us some confidence to believe that this must be true. Also, the work was done in Boston, so it probably must be true, right? So, but then we went down the list and we said, okay, what are the other genes that which could be targetable over here? And the topmost targetable gene in this list, quite to my surprise at least at that time, was a gene called BCL2L1. The protein product that this gene encodes is called BCLXL. You might know it as an anti-apoptotic protein. So we went and we started validating whether this result actually was true, and it was actually holding true in a number of different cell lines. We could inactivate BCLXL, and we found that unlike its very closely related sibling BCL2, loss of BCLXL impacted the growth of kidney cells much more profoundly. So on the upper left, as you can see, the black is the amount of cells that are surviving when we inactivate all of these particular genes over here. The controls, you see a lot more cell growth. BCL2 eliminates many more of these cells than BCLXL eliminates many more than BCL2 does. And this was on target because we were able to put back a resistant version of BCLXL and now see that the hairpins no longer worked, showing that the effect of the hairpins were actually on target. So what exactly might be going on over there, and how could we utilize this information from a clinical standpoint? There are great pharmacological agents available to target BCLXL, BCL2, and it was almost natural for us to go and ask whether the RCC cell lines would respond to these pharmacological agents. We found that BCLXL inhibition was indeed able to very acutely target almost 40% of the RCC lines that we tested in the lab. And this was again specific because the BCL2 inhibitor did not do anything to these cell lines. They survived happily in the presence of this drug. So what might be driving this difference? Why are 40% of the cells that are responding and why are the others not? We started doing some comparisons of these responders versus non-responders. So we did some RNA-seq analysis and we tried to look for gene sets that might be different between these two sets. And what we did find was there were indeed predictive markers over here. But again, to my surprise, epithelial to mesenchymal transition, a cell state conversion seemed to be one of the topmost gene sets that was marking these two different cohorts. Importantly, you can also use this predictive marker, and you'll see that about 30% of the human tumor samples are showing a signature that would correspond to BCL-XL sensitivity. So this might be relevant down the line. But more importantly, from a biological standpoint, I was more interested in asking whether this was a causal signature. And so what we found was you could take a cell line that was otherwise resistant to BCL-XL inhibition. You could turn it more mesenchymal, in this case by treating it with TGF-beta, and now it became sensitive to the BCL-XL inhibitor. So it was the conversion of cell state was conferring BCL-XL dependency, which again, what to me was incredibly surprising. And we started thinking, what might this mean? And I'm going to leave you with a very speculative idea. There's a pie in the sky sort of idea. Essentially, we think about shedding from normal epithelial structures, cells are falling off, but then they are immediately primed to die. So there's a phenomenon called anoikis, or it's Greek for homelessness. So everything sounds cooler in Greek. And so I think this is why we think about phenomenon by which our body automatically clears these cells out. A tumor cell, though, has to extravasate, go to distal tissue, and now that is a mechanism where it has to survive these cell death phenomenon. And that's what we think BCL-XL is doing in RCC lines. As RCCs become more mesenchymal, their dependence on BCL-XL also increases, and this is what allows them to survive this insult. So I'm going to stop there. The stars of this show really are my trainees, Treg Grubb and Smriti Maganti, and then a number of collaborators, including people in Bill's lab, at the Broad, and at the Harvard School of Public Health, who all helped along the way, and of course my collaborator at the Cleveland Clinic. And thank you to the funding, and that's the group of people that we work with.
Wonderful, thank you. All right, and our last speaker for this session is King Zhang from UT Southwestern, talking about targeting HIF-2 alpha inhibitor resistance in clear cell RCC.
Okay, all right. Are we good?
They're waiting on your slide.
You hear me? All right, it's kind of hard to talk after Abhishek. Such an impressive presentation. So today I might talk about some of the preliminary data we recently generated on trying to actually tackle the acquired resistance toward HIF-2 alpha inhibitor. So as we may know that renal cell carcinoma is characterized with loss of the VHL loss, which actually leads to the stabilization of HIF-2 alpha and transactivates HIF-2 alpha target genes. So MK6482, or so-called PT2399, and also later on called bazutifan, is one of the compounds that is clinically approved in the Journal Association to treat VHL-associated renal cell carcinoma. So in that sense, in the earlier study published Dr. James Berg-Wallace at UT Southwestern by using the patient-derived xenograft, in this case XP164. So you could appreciate that over long-term of PT2399 treatments, tumor originally will shrink, but over time, those resistant clones will develop, actually prolong, and then over time, those mice will succumb to the disease. So one of the mechanisms will be actually the domain that is mutated between HIF-2 alpha and HIF-1 beta, which was shown in Dr. Berg-Wallace's lab. But there also may be other mechanisms, either epigenetically or genetically, that could contribute to this process. So how can we actually address this question? So we decided we're going to generate two cell lines. So in this case, we actually treat CCR60 cell line 760 chronically with PT2399. In addition, on top of PT2399 treatment, we're also using CRISPR-Cas9 to totally abrogate HIF-2 alpha expression in those cells, which I won't show you here. So when you look at this background on the left, you can appreciate that for those sensitive cell lines, when you treat it with PT2399, you see that 70% of colony growth on the soft agar will diminish. But on the other hand, over the course of the 4 to 6 months culture conditions, we actually saw several resistant clones developed on 3D soft agar. So we were able to actually retrieve cells from those colony grown as a resistant clones, and then grow them in the long-term culture as a resistant model. So you can see on the right, this is the morphology showing that sensitive cell line will actually undergo the growth defects with PT2399 treatments. On the other hand, those resistant lines in several different versions showing some heterogeneity, but in any case, they're showing the robust outgrowth of the colony formation. So once we have this tool, what are you going to do with this? So we decided using a lot of genomic studies and epigenetic features to see whether we can tackle the molecular mechanism. So this put 1 year or 2 years work in one slide, where we actually perform ATAC-seq, look at chromosome accessibility. We also look at some of the epigenetic markers, including H3K27 acetylation, as well as H3K for monomethylation. Also, we combine with RNA-seq. We want to see actually on the top of the heat map, you can see there's increased accessibility for some of the resistant clones compared with parental lines, but also it's showing increased RNA expression. So the question is what kind of pathway that actually is potentially mediating this resistance? So presently enough, when we actually look at gene set enrichment analysis, so most robust pathways that was enriched is actually interferon, type 1 interferon response signaling pathways. So a lot of people will be puzzled because interferon signaling pathway is supposed to be actually is a promoter of the tumor immunity. But in this case, how could those interferon pathways be stimulated in those resistant clones? So one of the potential pathways could be that somehow they go rogue during this process of the resistance. So I just want to give one of the examples. One of the genes we actually found in signaling pathway, so-called P72. So you can appreciate in resistant clones, there's highly upregulation in P72 expression. Also showing in the middle, there's increased accessibility in terms of chromatin accessibility. Also on the western blot on the right, there's robust upregulation in this P72 proteins in those resistant clones. So functionally speaking, what we can do is that we can actually take those P72, overexpress them, and then we combine with HIF-2α inhibition. We want to see actually on the right, you can see those colony formation. In terms of HIF-2α inhibition, you can see parental cells respond to HIF-2α inhibition. On the other hand, if you actually overexpress P72, these actually cells will undergo resistance toward HIF-2α inhibition. So in conclusion, I just want to show you brief studies where we actually established some of the system for HIF-2α adaptive resistance, also showing some using multi-omics approach, showing potential pathways that may mediate this resistance. We also use gain of function as well as loss of function, showing that this P72 actually may be important mediating its HIF-2α resistance. So lastly, I want to thank the people who contributed work I actually acknowledged along the way, but also funding resources. Thank you so much.
So we have time for questions, for 10 minutes of questions for our panelists. Anybody in the room?
I have a question for Abhishek. I was really intrigued by your finding. So in some other cancers, for example, in breast cancer, typically EMT has been associated with cancer stemness and therapeutic resistance. So do you think it is quite specific for renal cancer and it's specific for BCL-XL inhibitor response?
When we originally found it, that's what I thought it would be. But turned out that after we published, Chris Wood from Duke, he reached out to me and he said that they had found in other lineages as well, there was a correlation with mesenchymal state and BCL-XL dependence. So I think this might, I don't think it would extend to every single tissue probably, but I do think that the evolutionary selection is not limited to the kidney alone. That's a really great point to think about, and I didn't have time to elaborate on that. But being more mesenchymal, these tumors are the more aggressive tumors in the clinic, right? And so what we are seeing over here is a potential target in really hard to treat tumors. And so we're really excited from a clinical standpoint that we might have a target to hit tumors that we are not able to target right now.
Dr. Zheng, outstanding presentation, thank you. Can you just tell us a little more about P72 and what is your proposed mechanism? I mean, so far it's a nice observation through the multi-omics. You start with RNA, you show on the protein level, you clearly show that reinduction of P72 confers the resistance. I think that for so many sessions so far that have shown our attempts to identify resistance to the Ipi-Nivo, can you just try to tell us, and in this case, for example, it's a belzutifan, what do we know about P72 and what is the proposed mechanism? Where do you think it acts? What do you know?
Right, I think that's a great question. The short answer is I don't know yet. And then if I have to speculate, I think this is epigenetic actually enhancer upregulation of the P72, as well as other related signaling molecules in type 4 interferon pathways. And then by upregulating those key genes in some of the interferon signaling pathways, and then cells somehow actually overcome this HIF-2 alpha inhibition or absence. In terms of detailed mechanism, we actually still undergo active investigation. So I don't have the answer for you. Hopefully next time we can have better answers.
Just to follow up, we have the Merck 5764 initial Peloton drug, the Merck that was just published, presented at ESMO. About 10 or 11 or 15 patients or so had a resistance. I think this may be your key to see if you can get samples from those, because there have been patients that came off due to the reasons people came off from the trial, or either toxicity or progression.
Yeah, so, excellent suggestions. I think the next one we'll actually try to find some clinical relevance, especially for those resistant patients over long-term treatment, to see whether P72 is upregulated as well. Great.
Thank you, Tony.
I want to follow on this. And,
yeah.
How are you going to follow by human samples of us to see how this are you going to do? I mean, if this is an epigenetic, marker, how are you going to follow on a human tissue? Are you going to go for IHC, look at the protein? What's happening in the modification, chromatin modification here?
Yeah, I think it's a couple of ways we can do this. So one is that we can look at, for example, the largely available patient-derived xenograft, where we can potentially manipulate those P72 gene expression, either overexpression or actually gene depletion, to see whether they could actually reverse some of the HIF-2 alpha resistance. On the other hand, we can also perform some of the epigenetic markers, including ATAC-seq and H3K27 acetyl for either paraffin-embedded tissues or actually other potential clinical samples. So that's actually these days actually pretty feasible to do, especially in patient samples. But we haven't done that yet. But I think that's a great suggestion.
Great talk. Did you identify any mutations in HIF-2?
Yeah, so there actually, I flew through that pretty quickly. So we particularly focused on HIF-2 alpha independent. So what we did is actually first, we actually knock out HIF-2 alpha completely, or actually use orange, knock out orange completely. And second, on top of that, we add HIF-2 alpha inhibitors. So even during this total absence of HIF signalings, either HIF-2 alpha, HIF-1 beta, you can still see that 30% of cells still possessed on 3D soft agar. And then after that, we actually do this treatment, do this outgrowths. And then we also perform the exome sequencing for those cell lines. We actually did not see a lot of mutations that caught our eye, so which actually prompt us to do some of the epigenetic gene expression change. So we don't think it's particularly mutation-based, but rather than actually gene expression change-based that is contributing to this phenomenon.
Back of the room.
Last question. So thank you for the talks. My question is about the transcription factor STAT3 that was discussed, and we have been working on this tumor microenvironment, and we found that the STAT1 and IRF1 are actually expressed, with its regulon on the peripheral of cancer nest. So, I was just wondering if you have noticed something like that for STAT1, IRF1. Are you planning to explore that? Because, our analysis, show STAT5, STAT1, and IRF1 are expressed on the border.
I haven't actually looked into STAT1 or any other stats. Just because we've just started this
just
particular.
Wait, they can't hear you. Check your mic.
Oh, sorry. That's a great question, and we haven't looked at STAT1, but we are planning on looking at that in the future, just because we've just started this project, and STAT3 is the main focus of our lab, and STAT3 inhibitors. But yes, we would love to look into STAT1 and the other stats.
Thank you.
Any other questions? Okay, we'll go on to the next
Thank you very much.
panelist.
Tumoral immunometabolic coevolution in clear cell. Oh, sorry. Oh, we got the thumbs up. All right, our last three here. So really interesting. Save the best for last, maybe? I'm just kidding. Again, we have Cerise Tang from Memorial Sloan Kettering talking about tumoral immunometabolic coevolution in clear cell RCC.
Okay, so thank you for the introduction. As mentioned, my name is Cerise Tang, and I'm currently a PhD student at Memorial Sloan Kettering. And today I'm going to be presenting on my projects, metabolic heterogeneity and the tumor microenvironment in clear cell RCC. And before I begin, I just want to thank the organizers here for giving me the opportunity to present my work today. Okay, so a little bit of background. We know that clear cell RCC is driven by a loss of function mutation in the VHL gene, where a lot of the downstream pathways that are modified are metabolic in nature. But recently, two new phenomena have kind of changed the way that we looked at clear cell RCC. And the first one is that we know that clear cell RCC is very genetically heterogeneous. So previous work has shown through multi-region analysis that there are somatic mutations and chromosomal imbalances that differ between a tumor, leading to phenotypic differences intratumorally. And also when they did evolutionary phylogeny, they found branch evolution, which shows that there are multiple subclones that are co-evolving within a tumor at the same time. And the second new phenomenon that has really changed the way we look at clear cell RCC is knowing that it's highly immune infiltrated and that it responds really well to immune checkpoint blockade as well as combination regimens. So knowing that clear cell RCC is metabolically driven, genetically heterogeneous, and highly immune infiltrated, it led us to ask two different questions. The first is, are clear cell RCC tumors metabolically heterogeneous? And if so, how does it manifest within a tumor? And secondly, do metabolic changes co-evolve alongside changes in the immune microenvironment? And in order to answer these questions, we put together a cohort of 31 multi-regionally sampled clear cell RCC tumors that had metabolomics, RNA sequencing, and whole exome sequencing performed on them. So the first thing that we did was we just did a basic Pca analysis on our metabolomics data. And predictably, PC1 separated the tumor samples from the normal samples. But when we looked at PC2, we saw that it separated the tumor regions from each individual patient. So seeing the separation across PC2, we classified our tumor regions as being either PC2 high or PC2 low. And then it's important to note here that every tumor had PC2 high and PC2 low regions. And then when we looked at the differences between PC2 high and low regions metabolically, we saw that there were differences in expression of cysteine and glutathione and other other metabolites, and then also polyunsaturated fatty acids, which are labeled in green here. So what is the molecular phenomenon behind these metabolic changes? And we think that it has to do with ferroptosis. So pretty much what happens in ferroptosis is when you have high ROS levels within a cell that exceeds a cell's antioxidant capacity, it leads to the oxidation of these polyunsaturated fatty acids. And when these polyunsaturated fatty acids are oxidized, it leads to the cell being signaled to undergo ferroptosis, which is a form of iron-mediated cell death. However, when you have high expression levels of antioxidants such as cysteine and glutathione, that can reduce these oxidized polyunsaturated fatty acids, leading to the ferroptotic switch being turned off. So what we hypothesize here is that this pattern of intratumor heterogeneity that we see across these PC2 high and low regions is compensating for apoptotic susceptibility. The next thing that we wanted to look at was the interaction between the metabolome and the immune microenvironment. So what we did is we used a mixed effects model to look at the relationship between the metabolites and an RNA-based signature of immune infiltration. And what we found was approximately 5% of the metabolome is significantly associated with this signature, including metabolites in the NAD metabolism pathway. And one of these metabolites is known as nicotinamide, and we see that when you look at within a specific tumor, the regions that are highly immune infiltrated have higher levels of nicotinamide. And then more generally, when we looked at the myeloid signature, which is a prognostic signature for progression-free survival with immunotherapy, we found that there are also a bunch of metabolites that are associated with it with a mixed effects model. And this work is still in progress. So yeah, in conclusion, within a single clear cell RCC tumor, we find that there's a dominant heterogeneity program that is driven by polyunsaturated fatty acids, cysteine accumulation, and ROS tolerance, which we hypothesize is in response to apoptotic susceptibility. We also found that in clear cell RCC, NAD metabolites, specifically quinolinate and nicotinamide, are associated with higher immune infiltration intratumorally. And the broad overarching conclusion of my work is that metabolic heterogeneity is pervasive in clear cell RCC and can be remodeled by the presence of immune cells. And with that, I'd like to thank the Resnick and Schultz labs at MSK and the Krumschick lab at Weill Cornell, as well as the medical oncology and Department of Virology at MSK for their assistance. Thank you.
All right, our next speaker, switching gears a little bit, is Kevin Hakimi from UC San Diego, discussing an interim analysis of the Padres trial.
Thank you. Thanks for having me here, everyone, and thank you for the introduction. Today, I'll be presenting the interim analysis of the aptly named Padres trial. The study is funded by Pfizer, and I have no conflicts of interest to disclose. Up to 25% of renal masses present with CKD or other imperative indications, such as solitary kidney or bilateral renal masses for nephron-sparing surgery. Patients with large and complex masses nowadays are increasingly able to undergo partial nephrectomy, but there are a subset of patients that aren't able to undergo the procedure because of lack of adequate oncologic outcome and renal insufficiency post-procedure. In the same light, pre-surgical therapy with neoadjuvant TKIs have shown promising results in reducing disease burden, but haven't shown to facilitate partial nephrectomy in any dedicated clinical trials. The aim of our clinical trial is to assess the use of pre-surgical TKI with zidenib and assess the feasibility of this neoadjuvant approach to facilitate partial nephrectomy in circumstances in which that procedure may not otherwise be feasible. This was a single-arm phase 2 study using zidenib in patients with localized clear cell renal cell carcinoma with imperative indications for nephron-sparing surgery. Our inclusion criteria included patients with an eGFR of less than 60 or a functional solitary kidney, a renal nephrometry score of greater than or equal to 10, or radical nephrectomy in these patients would lead to an eGFR of less than 45. AMPT exclusion criteria includes patients with metastatic disease, a non-clear cell histology, or prior treatment for their RCC. To evaluate each of these patients, each patient had cross-sectional imaging to delineate the renal mass and surrounding structures. They had labs drawn to determine creatinine-based estimation of GFR. They had metastatic evaluation with a chest CT, other scans as amply noted. They had biopsy to confirm the clear cell histology, and tumor complexity was defined by renal nephrometry score. Patients were enrolled and received a zidenib 5 milligrams PO BID for 8 weeks and titrated up as needed. And outcomes were assessed, including tumor response after completion of neoadjuvant therapy, the ability to perform partial nephrectomy after our therapy. Functional issues such as avoidance of dialysis and severe CKD was also indicated, and the avoidance of major surgical complications, mainly Clavien greater than or equal to 3 complications, was also assessed. Looking at our patient and tumor characteristics initially, you could see that all of our patients met criteria for study entry, either with an eGFR of less than 60, a solitary kidney, or a renal complexity score of 10 to 12. All of our patients received 8 weeks of zidenib with no grade 4 or 5 complications. 5 patients had grade 3 complications. All were adequately adequately treated with medical therapy and able to undergo the total 8 weeks of azitinib therapy. Regarding the post-azitinib tumor characteristics, azitinib resulted in a significant reduction in median tumor size of about 19% from 7.7 centimeters pre-treatment on average to 6.3 centimeters pre-treatment post-treatment on average. Azitinib also resulted in significant reductions in median renal score, with the main determinant of outcome in the renal score being size of the renal tumor or the radius or the location relative to the hilum. And ultimately 9 tumors were downstage, and I'd like to point out that most of the tumors that were included in this trial were T3A or greater. Ultimately, 25 out of the 26 patients enrolled in the trial were able to undergo surgery, with 19 out of those 25 patients able to pursue partial nephrectomy, not being able to pursue partial nephrectomy initially. There were 6 grade 3 or 4 Clavian grade complications, 4 of them being urine leaks post-procedure, and 24 out of the 25 patients that ultimately underwent surgery achieved negative margins. Regarding renal functional outcomes, here's a graph of the percent change in eGFR at 1 month post-op for these patients. 2 patients achieved a greater than or equal to 50% reduction in their eGFR following surgical resection of their tumor, both of those patients undergoing a radical nephrectomy, and 1 patient had long-term dialysis dependence, that patient also undergoing a radical nephrectomy in our trial. The median eGFR change between pre-azitinib and 1 month post-surgery was about 19%, and while statistically significant, looking at the values, not very clinically significant. These patients were able to avoid dialysis dependence, were able to avoid a significant drop in their eGFR following our procedure. So the key points and conclusions of our trial: this neoadjuvant method may safely and effectively downsize tumors and facilitate partial nephrectomy in patients who are not able to undergo that procedure initially, and may allow patients to avoid complications such as dialysis dependence long-term. And thank you to the patient and their families, and I would like to thank all the mentors who are involved in this trial, Dr. Rana McKay, Dr. Ithar Darwish, Dr. Rini, and Dr. Campbell. Thanks so much.
All right, and our last presenter is Anupama Reddy. So we're again shifting gears one more time, talking about machine learning in the OPTIC trial.
Hi, good afternoon, everyone. My name is Anupama Reddy. I'm a data scientist and a bioinformatics geek. So I'm very excited to present this collaborative work with Vanderbilt University Medical Center on machine learning modeling using RNA sequencing data.
There you go.
So our project is on machine learning modeling using RNA sequencing data for patient stratification in a prospective phase 2 clinical trial. Okay, so based on data from Emotion 151, where they ran RNA sequencing on over 800 patients, they very nicely identified 7 molecular clusters, which were associated with distinct biological processes. So cluster 1 and 2 was associated with angiogenesis and very nicely showed responses to TKI inhibitors. Clusters 4, 5, and 7 showed response to IO therapy. And so based on these results, so Dr. Rini and his team at Vanderbilt are setting up this innovative trial called OPTIC. There's also a poster right on that wall presented by Dr. Hake, which describes this trial in more detail. So the goal is to test the clinical efficacy of this RNA-seq-based patient stratification. And so we were tasked with developing an accurate machine learning model that can assign patients into these groups. So, one of the main translational challenges that we had for this model was that this was a prospective clinical trial. So we were going to make predictions on 1 patient at a time. And that's a problem for normalizing gene expression data. The commonly used techniques are z-scoring or some kind of batch normalization, which we couldn't do because we just had 1 patient, right? So we had to kind of think of other innovative solutions. So the solution we came up with was we would normalize in the gene space we would compare pairs of genes and take gene expression ratios. So the main difference is that instead of saying that is this gene high or low, we would say is this gene greater than the other gene, right? So and this method also very nicely translates when you're using when the assay changes between your training data set and your test data set. As a bonus, it can even improve kind of some of your, it can improve the accuracy. Okay, so in terms of the methods, right? So the data that we used, so we used the Emotion 151 data, where we had 823 patients, which were mostly CCRCCs. We excluded cluster 7 because there were only 28 patients in that cluster. We used the log2 gene expression values, and we grouped the clusters into 3 groups because that was that's how we were going to stratify the patients into the arms. So cluster 1 and 2 was angio, 4 and 5 IO, and 3 and 6 we would exclude them, but we still had it as an outcome that we were going to predict. So in terms of the model, we selected features, we computed all pairwise gene ratios, and then used random forest for the multi-class classifier. And for validation, we did cross-validation, which is randomly splitting your training and test sets multiple times. And then we used metrics such as accuracy, sensitivity, and positive predictive value. We also looked at some independent metrics, which were not based on the true label. And we did an independent validation in a cohort of 65 samples. So in terms of the cross-validation results, so we see an overall accuracy of over 85%. This is results in cross-validation where it was repeated multiple times. When we look at sensitivity and positive predictive value for the 3 groups that we were going to classify the patients into, we see that it's very high, it's over 80%. And cluster angio, it's even over 90%. So we were able to very accurately predict patients into the cluster IO group. When we looked at the independent metrics, so this was looking at predictions versus survival or clinical response, then we also, the groups, the results lined up very nicely in terms of the prediction. And in terms of validation on an independent cohort of 65 CCRCC patients, so this data was provided by Dr. Katie Beckerman, also at Vanderbilt. So we see that majority of so this is a heat map here showing 65 patients along the rows, and along the columns you have genes which are associated with one of the 7 clusters. And this is gene expression, these are gene expression values. So red is high, blue is low. So what we see is that majority of the patients ended up in the angio cluster, just based on this one sample of 65 patients. And then the IO was a much smaller cluster. But we see that the patterns of expression of these genes, very nicely line up with the predictions. And when we split the predicted samples in each of the clusters by their treatment, whether they were treated, so for example, the angio, the predicted angio group, whether they were treated with IO and TKI or just a combination of IO, we see higher proportion of responders in the IO and TKI, and this was statistically significant. We also see a similar trend in the predicted IO group, but the sample size was very small. And our goal is to, add additional samples to this cohort. So in summary, we have a good working machine learning model, and the novelty here is we're using gene ratios. It performs consistently in an independent validation set, and we've also developed a software platform that can automatically, it's cloud-based and can automatically process the data and give the predictions. And yeah, I'd like to thank all of you for your patience.
Questions?
We've got about less than 5 minutes for questions for last panelists.
Jen.
Thank you. So this is about the neo or pre-surgical therapy. So if you were to design the PADRES trial today, would you be using an IO-TKI combination, given, for example, that response rates with lenvatinib combinations are 70%?
Are we good? Okay. That's a great point. I think the next steps, this is an interim analysis where our plan is to enroll patients up to 50 for this trial specifically, but I think where immunotherapy is going, especially in the treatment of RCC, that's kind of the next step to see whether these patients would benefit from any IO treatment. Whether it's in combination with TKI is still yet to kind of be officially promulgated, I guess. But I think the next step is really to see whether this can be used in a broader application for patients and not just in patients that have these imperative indications such as a GFR of less than 60 or a solitary kidney, but also whether immunotherapy would work better in reducing the size of these tumors and kind of preserving more renal parenchyma.
Well, fantastic talk. I have one question for the machine learning process. One thing that makes me really nervous whenever you're working with RNA is the potential that you will have some RNA degradation during the process of collection. So within this process, like going one by one sample, how can you control and try to avoid like those potential problems in your model?
Yeah, you're absolutely right. That's our worry too, especially with FFPE samples and so on. So the assay that you use can help with that, right? And we're currently, Tempus is our sequencing provider, and they use an exome capture-based RNA sequencing method, right? And that has been shown to work well against RNA degradation. And we've also performed some, some direct comparisons where we have run some samples with that platform, with the Tempus one, versus just with total RNA capture, and we see that we get better performance with the capture method.
And then to piggyback off that, how will this eventually be ready for prime time, as we say, in the clinic for this type of, putting these information in for our patients and utilizing this? When do you think?
I think the results from the trial will tell us. So the phase 2, we're so excited. I think it's going to start very soon.
Or what's like the timetable from patient getting diagnosed to getting the tissue, doing the sequencing, and you spitting out a test result? What's sort of your...
Yeah, our hope is 10 to 14 days.
Nice.
It's pretty quick.
Any other questions? All right, last question. Oh, sorry, we're looking at two different. Martin, you want to go?
Yeah.
And one more over here.
Machine learning, that was a great talk, thank you. So your distribution is heavily slanted towards the angio in your data sets. From the original cancer cell paper, we know that distribution very crudely depends on the risk grouping, IMDC, intermediate poor. Do you have a way to look at your distribution, match the makeup of your patient cohort, and see how well you recapitulate what was seen on 151?
Yeah, it was surprising to us too that we saw, majority of the patients, but it could just be this sample set. But you're right, we could look at the distributions and compare them to the mod survey.
So how well do you match ultimately with the trial data set in your models, your cohort?
So we haven't done that comparison.
We're done.
Well, thank you so
much. Oh wait, there's one last one. Last minute.
Okay, my question was about the metabolic states that was discussed about the ROS and OXPHOS and fatty acid. So I have a question first about connection between ROS and OXPHOS. Is there any causal relation between them? Have you explored that? Also, having more ROS, gets this cancer cell more oxidative, phosphorylated, have that pathway more active. That was the first question. The second one was about prognostics of these pathways. Have you ever, noticed something that, OXPHOS cancer cells have more, worse prognostics? Have you explored that? And the third one was, but fatty acid metabolism.
We're at time.
Okay.
I'm sorry. So have you, checked like fatty acid oxidative and also fatty acid synthesis? Is there any difference between them? Have you explored that one also?
Okay, I'll answer the last two. So when we did our like GSCA, we didn't see any differences in like rates of fatty acid synthesis. And in terms of prognosis, like we haven't looked at anything related to like reactive oxygen species, but we do think that the finding is still interesting because I think ferroptosis is like kind of like a new phenomenon, and like people are trying to like target it with drugs. And what we're showing is that within these tumors, there are going to be some regions that are not going to respond to these like ferroptotic drugs, just because they have these like compensatory like mechanisms to like increase antioxidant expression and like these higher cysteine levels. And if there are some regions that will like compensate against these ferroptotic drugs, that means that they just will not like, what's the word? Yeah, they just won't respond to these drugs. Yeah, not in terms of ROS specifically, but in terms of the ferroptotic pathway, that's what we hypothesize.
Thank you. Thank you all for a wonderful session. You guys were all great.
Thank you so much.